I'm happy to welcome Zarmeen Zahid with her talk Psychedelic Induced Plasticity and Metaplasticity in Murine Hippocampus. She is a fourth year PhD student at the University of Wisconsin-Madison. And besides her PhD work, she's also a science communicator and co-founder of the multimedia platform Your Brain on Science, where she wants to bring or make psychedelic science policy and culture more accessible and understandable for the general public. Warm welcome. And yeah.
Thank you so much. Congratulations on making it to the last presentation of the last talk. It's been a really long day, but an incredible conference. So thank you to everyone who helped get this set up. So I am now a fifth year PhD student, hopefully done soon, in the labs of Matthew Banks and Cody Wenther. And I'm going to be talking to you today about some of my thesis work having to do with psychedelic induced synaptic plasticity and metaplasticity in the murine hippocampus and the mPFC, but you don't see that up here, and I'll show you why in a second. So let's get into it. Why are we all here? It seems like a silly question to ask at the end. But as we all know, in recent clinical trials, serotonergic psychedelics, and I'm going to talk specifically about serotonergic psychedelics because that's what I did my work in, and I use that to say that these drugs are active at the 5-HT2A receptor, right? So a ton of psychedelics have been very effective, very efficacious in treating very intractable disorders such as depression, anxiety, substance use disorders. And if you've sat through these talks yesterday, you know that there's a lot more. There's many more indications that these drugs are actually being tested for right now. So this list will most likely get longer. And the literature suggests that these changes that we see are rapid and very long lasting. And in the case of depression, for some patients, the effects of psychedelics actually outperform that of antidepressants. And talking about the STAR-D trials, which were a very comprehensive sort of overview of efficacy of antidepressants, we find that only in 67% of cases did patients actually achieve remission. And in a recent reanalysis, it suggests that that number might actually be smaller. So these sort of results that we're seeing with these psychedelic trials are highly impactful for a lot of patients, clinicians, and scientists sort of alike. Despite this, really exciting, I do want to point out that there's a gap in our knowledge. The mechanism by which these behavioral effects are sort of seen, the therapeutic effects are seen, is still very unclear to us. And I want to point out that a lot of these clinical trials are psychotherapy linked, right? So these are psychotherapy assisted paradigms. So there's really this inextricable link between the therapeutic outcomes and the therapy itself, right? So this is actually a therapy room over at UW-Madison, and it's currently being used in, I think, the RECAP trials and a couple of other really interesting things that I would love to talk to you about if you're interested. But this points out a challenge with clinical work, right? So we are unable to really separate those biological changes that are occurring in response to drug administration versus those psychological changes that might be occurring due to that psychotherapy component, right? It's really, really important. So here is where I'm gonna talk about why sort of preclinical research is really important, because this is where we come in. Here, we can use rodent models to sort of isolate those biological effects to the best of our abilities, right? Isolate those biological effects and understand at the circuit level sort of what is happening. And in a lot of clinical trials, of course, there was EEG and fMRI, and that's gonna tell us something about global dynamics, global functional dynamics, changes in the brain globally. But again, that circuit level mechanism is still sort of unknown to us. So in my work, I use rodent models to help us sort of understand really what is happening. And I'm gonna hone in to one hypothesis in the field that we talked quite a bit about this morning if you attended Dr. Goldolan's talk and the several other talks. So I think you guys have heard this quite a bit today, but there's this hypothesis in the field, and it's the pro-neuroplastic hypothesis of psychedelic action, which posits that psychedelics may be inducing their behavioral effects through their action as psychoplastogens. What are psychoplastogens? Drugs that induce neuroplasticity. Heard that all day. What is neuroplasticity? Neuroplasticity is the brain's capacity to prune, form, and reorganize in a way that is optimally efficient, right, to deal with whatever is happening and whatever change might have sort of occurred. So there's actually evidence that psychedelics might induce this plasticity. And I'm saying plasticity, we'll get a little bit into the weeds about plasticity and metaplasticity. But there's evidence that psychedelics induce plasticity in a couple of ways, and my citations are no longer comprehensive. There's a lot more data here. But at the level of transcription, there's evidence that single administration of serotonergic psychedelics actually induces the transcription of immediate early genes or gene plasticity-associated genes. So things like your AMPA receptors, your NMDA receptors, and BDNF. And your AMPA receptors and your NMDA receptors are going to be really vital in that early phase of plasticity of LTP, which is a measure that we use to see plasticity. And BDNF is going to be, heavily involved in that robust form of long-term sort of plasticity. So at the level of the synapse, we see that there's synaptogenesis. There's evidence for synaptogenesis, so an increase for the number of synapses and sort of the synapse size. Further, there's evidence for spinogenesis. So those are those tiny little sort of projections coming off of those dendrites. We find that after administration of serotonergic psychedelics, there's an increase in spine size, stability, and perhaps spine head diameter. There's quite a few studies showing that. There's evidence for neurogenesis, so actually the creation of new neurons in a few cortical areas. And then there's evidence for functional plasticity. So this is going to be about the functional dynamics of those cells, right? How they're talking to each other. There's evidence that serotonergic psychedelic administration is going to be associated with lower thresholds for action potentials. And in Dr. Goldolan's talk, we saw that there is also LTP and LTD associated with serotonergic psychedelic administration. Now, I'm going to take a moment to address plasticity and metaplasticity. And for plasticity, I'm going to read, let's read directly from this quote. Plasticity, as we just talked about, is the ability of the nervous system to change its activity in response to intrinsic or extrinsic stimuli by reorganizing its structure, function, or connections, right? So that's what sort of we just talked about. Metaplasticity, on the other hand, is manifest as a change in the ability to induce subsequent synaptic plasticity, such as long-term potentiation or depression. So metaplasticity is really the more robust change that we're looking for, and it's almost, the way that I think of it, it's plasticity of the plasticity. How much can you modify the plasticity that you've seen that's occurred? And we heard the term, hyperplasticity, and that might be appropriate to use in some cases, but metaplasticity is really sort of what we're looking for here for long-term sort of enduring changes. So now that we have that, we sort of need to identify a brain area that we're going to look in. So I'm going to talk about the hippocampus and the medial prefrontal cortex, and also put these two areas together and look at it in a circuit. So the hippocampus, as I think we all know and are familiar with, plays a very big role in learning and memory. And it also has an immense capacity for plasticity. So the circuits here are highly amenable to structural reorganization based on some learning or memory event. And the hippocampus also modulates brain oscillations and neurotransmission in response to learning and memory events, so a pretty vital area. So now the prefrontal cortex is vital in higher order cognitive functioning, so things like decision-making, reward valuation. It's going to be involved in emotional regulation, having to do with its reciprocal connections with the amygdala and so on and so forth. Now let's put these two areas together. So there is actually a direct circuit from the hippocampus to the medial prefrontal cortex, the ventral hippocampus, there should be a V there. And circuit level dysfunction in this circuit is actually noted in various conditions. So in humans, so in individuals that are suffering from depression, you'll see weakened connectivity in this circuit. And what's really interesting for me and really important for me is that this circuit is also reliably dysregulated in animal models of stress and acute threat response. So this tells me that if I try to look at this in a rodent model, that I might see a signal there, right? So now at the level of behavior, because eventually we're going to work our way up to behavior. This circuit is also implicated in various behaviors, and I'm going to kind of fly through this. But the novelty suppressed feeding task, which is a behavioral assay in rodents of positive valence, and it probes approach and avoidance behavior. So approach and avoidance behaviors are going to tell us something about anxiety, right? Anxiety-like behaviors, because we are looking in rodents. We can't say that the rodent has anxiety or depression, so we want to be very careful with our words there. Fear conditioning and extinction, which I'm sure a lot of you guys are sort of familiar with, which is a behavioral assay of negative valence that probes threat responding. And this, a lot of clinicians use, a lot of preclinical scientists use as a translatable sort of assay for PTSD. But this is really gonna sort of test the threat responding of those animals. And finally, cognitive flexibility. So cognitive flexibility refers to the ability to adjust your mental set based on whatever demands you're being given from the environment, right? So if you're given feedback from the environment, you know that maybe you need to change your behavior to sort of meet that demand or whatever is being asked of you at the time. And cognitive flexibility can be thought of as a form of behavioral plasticity, right? Being able to sort of respond and adjust in a way that is optimally efficient. So this circuit, it's a really good place to look. It has direct afferent connections implicated in various behaviors, and also has the brain areas themselves are also implicated in a lot of functions that we're interested in. So this makes us well equipped to ask the question, this is the question of my life, what is the role of synaptic plasticity in the mechanism of action of serotonergic psychedelics in pathways relevant to psychiatric disorder pathophysiology? And I hypothesize that administration of serotonergic psychedelics will increase plasticity and metaplasticity of cells in the ventral hippocampus to medial prefrontal cortex pathway to post-acutely affect long-term behavioral change. So there's a lot of pieces in this hypothesis, a lot of work, a lot of my time, but we're just gonna talk about this first piece where we're gonna look at plasticity and metaplasticity. So the experiments, I conducted 2 sets of experiments, that should say ex vivo. So excuse that, pretend like it says ex vivo. But we're gonna look at 2 sets of experiments here, ex vivo and in vivo. So let's start with the ex vivo results. So here, animals were injected, so C57BL6J mice, so I use mice here, were injected with either 1 mg per kg of DOI, and DOI is a serotonergic psychedelic that's selective, more selective for the 5-HT2A receptor, 10 mg per kg of ketamine, which is an NMDA receptor antagonist, and we use this as sort of a positive control here, and then saline, which is going to be our control condition. Animals were injected, and then at 24 hours, they were sacrificed, and cells were obtained to clearly visualize the hippocampus here. And what we did was lowered a recording electrode and a stimulating electrode in the Schaffer collaterals, the stratum radiatum portion of the hippocampus. The first thing we did was obtain stimulus response curves, and I'll show you guys what that looks like in a second, but basically you apply stimulation of varying amplitudes and then you record the response to that stimulation. Afterwards, we collected baseline responses to 0.05 hertz of stimulation, and this is gonna tell us something about the resting sort of functional dynamics of the cells that we're recording from, right? Like what do they look like as they respond without any induction, anything else? Then, using a theta burst stimulation protocol, we induced LTP and then again recorded at 0.05 hertz to see now if that LTP was induced or if there was a change, and really that's where the interesting stuff is gonna come from. So let's look at it. Okay, so let's start with this graph on the left here. So the x-axis is time. So we start at negative 30 minutes. Our baseline condition was 30 minutes long. So here is the responding at baseline. And again, I want to remind you guys, the drug is no longer in the system. 24 hours before we're doing these measurements is when the animals were injected with those drugs, right? So there's no drug in the system. And LTP is canonically measured at sort of the end of the hour. So at 50 to 60 minutes, I took the FEPSP slopes and sort of compared them across the conditions. And as you can kind of tell by looking at it, there was no difference. There was no difference in the LTP here between the saline DOI and ketamine groups. But I want you to keep this in mind, right? There's a few different things here that I hope you guys are seeing, but we'll come back to it. So I didn't find evidence for LTP here. So I talked about those stimulus response curves where we applied stimulation at varying stimulus intensities here and then recorded from those cells the mean FEPSP slope. And by eye, you guys can tell that there might be something here, right? So I took these data and fitted them to oops, don't know what that block is. You guys have to use your imagination a lot in my talk, and I'm really sorry, but these data were fitted to a sigmoid fit, a sigmoidal curve. And I promise you, even though you can't see it, these data were fitted pretty well. And what we could do here is then we can extract a few different parameters. So all the way on the left is S50%. So this is going to be the amplitude that produces 50% of the maximal response. M here is going to be our slope, and R max is going to be the maximal response that was evoked. So we find that though S50% and R max are not different, the slopes of the DOI group are significantly increased as compared to ketamine, right here, ketamine and saline, where ketamine and saline were not different from each other. So this is interesting. So this is telling us that in these stimulus response curves, there's an increase in the slopes after serotonergic psychedelic administration, right? So this is going to be evidence for plasticity, perhaps hyperplasticity, right? So that early form of plasticity. There's perhaps a change in the way that the cells are responding, right? So very small change here. We also collected some more data. So we have some fiber volley amplitudes. And fiber volley amplitudes are going to tell us something about the action potentials firing on the presynaptic side, right? So we just looked at some postsynaptic data, but this is going to tell us something about differences in the presynaptic side. And we find here, by comparing the fiber volley amplitudes across the groups, that the DOI fiber volley amplitudes were actually significantly increased compared to the saline group and the ketamine group. So this starts to hint at us that there might be something happening presynaptically. Now, there's a lot more that I can do here with these fiber volley amplitudes, and that is in the works, and I wish I had those analyses for you guys, but now we have an idea that maybe something presynaptic is happening. So now let's come back to that LTP figure that I showed you. So we looked at 50 to 60 minutes, right? But now let's look at this initial component, because this looks interesting. So when we look at 0 to 5 minutes, when we compare the slopes at 0 to 5 minutes, we actually find that the DOI slopes are significantly increased compared to ketamine and saline. Now here, because this is occurring in that initial portion, this is going to be evidence for short-term plasticity. And indeed, short-term plasticity actually refers to a phenomenon in which synaptic efficacy has been modified on the presynaptic side, right? So we got a hint of that with the fiber volley data, and now we're seeing a little bit more of that here. So this is actually going to be evidence for a change. It implicates changes in metaplasticity, right? Because although metaplasticity is more commonly thought of as a change in LTP or LTD, right, where we would see that here, STP is also a component of this entire curve, right? So this is going to be evidenced by a change in metaplasticity and presynaptically. So what do we see here in our ex vivo experiments? We see that there is an increase in cell excitability, so we have evidence for plasticity with our stimulus response curves. And then we see that there's an increase in short-term plasticity, and that's going to be our evidence for metaplasticity. So that's at 24 hours in ex vivo after injection of serotonergic psychedelic DOI. So now let's transition to the in vivo data. So here, there's quite a few time points. So 5 days before, 7 days, before any measurements are taken, I conduct a stereotactic surgery in which I implant a recording electrode in the medial prefrontal cortex here, and a bipolar stimulating electrode in the VCA1 of these rodents. So here, we're getting at that circuit, right? The VCA1 to mPFC circuit. So again, stimulating electrode in the VCA1, recording electrode in the mPFC. That's going to be important as we go forward. And we gave mice psychedelics here, right? So you might ask, Zarmeen, how do you know that these mice are experiencing anything, right? So our assay for psychedelic action in mice, so psychedelic onboarding in rodents, is something called the head twitch response. And the head twitch response is this really high frequency head movement. I'm gonna see if this video plays. This really high frequency head movement in which the rodent kind of like imagine a dog shaking off water or something like that. It's a very fast head shake, and you need special sort of cameras and equipment to see that. I don't know if you guys missed it. I'm gonna play it again. Ready? So it's that rodent right on the left. You see that head shake? So that's gonna be the head twitch. So we don't have high-speed cameras like that. So what we do is we created a magnetometer. So these are our cages wrapped in magnet wire that are gonna give us a signal like this. And because we're able to sort of isolate that high frequency sort of movement, we can take out those head twitch events right here. And that is gonna be our assay of, did these animals actually get the drug? Is it actually in the system? And there's a lot of controversy about this head twitch, and we can have a discussion about it. So the methods here were at baseline. So there's a baseline day where the animals are recorded from, and there is sort of nothing happening, just recording from the animals, spontaneous local field potential activity, seeing just how their cells respond normally. So on an experiment day, what we do is we take a baseline recording, so 30-minute spontaneous recording of those LFPs again. I conduct a stimulus response curve, after which the animals are injected, and then there's again a 4-hour spontaneous recording. And then these first two things happen again every single day for a week after psychedelic administration. And the drugs that were given here were saline, psilocybin in 2 different doses, 3 mg per kg, 1 mg per kg, 4-ACO-DMT, which is also a serotonergic psychedelic, and it actually converges on psilocin in the body. So this dose at 1 mg per kg should be comparable to this 1 mg per kg dose here with psilocybin. And then 6-fluoro-DET, which was given to us by our friends at USONA, and this is a non-hallucinogenic 5-HT2A agonist, and it is a DMT analog. So pretty interesting. We thought we would see what happens here. So the first thing that I did is assess whether or not these animals actually got the drug. And you can see here after time 0 that it does look like those head twitches increase, and then they sort of come back to rest over time. Here's an example of that stimulus response curve that I talked about. So this is what one of those responses in the medial prefrontal cortex in response to stimulation in the VCA1 actually looks like. This is a really good example from one of my animals. And we stimulated going from 0 microamps all the way up to 400 microamps and recorded the responses to that stimulation. And what did I find? So at the time of writing this talk and sending in my abstract, I had a really, really great result where my psilocybin group, my 4-ACO group, here we're looking at, fractional change of the maximum evoked response in baseline, because this was sort of the first thing that we looked at. We noticed that the maximal response looked to be different. And we found that the maximal evoked response had been different, but then I collected more data. And as I collected more data, this response really got drowned out here. And here with the 4-ACO-DMT and the 6-fluoro-DET, it kind of looks like there might be something, but I just suspect that if I continue to collect more data, that this will also likely sort of regress back to this mean. So really sad, but this is interesting, right? So also I will say that these data were also fitted to a curve, and we also saw nothing there, so I didn't just want to put all those graphs up here. But the results here with the in vivo data, and I mentioned that I take measurements every single day for 7 days after psychedelic administration. The results are the same, unfortunately. We also actually do this at 4 hours after psychedelic administration, and we, again, don't really find a signal there. So the results here, no plasticity, no metaplasticity observed. So to sum up what I have here, we find that ex vivo, there is evidence for plasticity and metaplasticity at 24 hours. But in vivo, I don't really see a signal. We don't really see any change here along this pathway. So what does that mean, right? What does that mean? So I am looking in this circuit that is canonically associated with anxiety-like behaviors. But it's really important to remember that my animals it was context devoid. There wasn't anything associated with the psychedelic administration, right? These animals were given the psychedelics, and then I took these measurements. Further, it's really important to think about the fact that, the brain has a lot of reciprocal connections, and especially the circuit between the hippocampus, the medial prefrontal cortex, and the amygdala. It's going to be very, very important in sort of the functions that we were talking about and the behaviors that we were interested in. So change in plasticity could be happening really at any part of that circuit. And perhaps that's just not the part of the circuit that I was looking at. But I think this adds to the body of literature. And that is all I got for you. So if you enjoyed hearing me talk, I do have a podcast and a website. Please listen to me more. If you didn't enjoy me talking, I'm really sorry, but it's done.
Hi, thank you for your talk. Suppose we did see an increase in plasticity. How good are our measurements for the increase we need to see in order to correlate it with therapeutic outcome? Because just spotting an increase that is statistically significant doesn't mean it's going to be therapeutically significant.
Great question. So that brings me to the second part of my thesis work, which is going to be looking at behavior, right? At the same time points, at the same time course that we were looking at these changes in plasticity. So the next steps for me are actually conducting those behavioral assays that I kind of flew through in the beginning and correlating those with the changes that I saw at 24 hours.
Hey, so I was looking at your head twitch graphs with the 6. Do you see head twitches with 6-fluoro-DMT?
So you shouldn't, right? But it's interesting to note that there is like a marginal increase here, right? And something important that I want to point out that you guys wouldn't know unless I told you, right? My animals are fitted with this ginormous head cap after I do these surgeries. And you take them out of the cage, you inject them, you put them back in, you've kind of ruffled their feathers a little bit, right? And their head caps are something that they hit at all the time. Although that doesn't really tell us why, the 6-fluoro-DET is a little bit higher than the saline, but it's not much higher.
So you see
than
the
saline. Yeah, exactly.
Okay,
so
yeah.
the blue is going to be that saline bar right there. But really good catch. Yeah.
Hi, thanks for the great talk. So combining everything we heard here in this room today, so like the very first talk from Dolan, but also the TRKB receptor, like when we talk about metaplasticity, there always seems to be the need for something that guides the neuroplastic change. And as you already pointed out, like that seems to be nothing really that intrinsically would guide that process. Might that be something you need to consider in order to actually find it in specifically the circuitry you look at?
Great point, and that's exactly why I mentioned it, because it was context devoid. I wasn't doing any behavioral assays. I wasn't introducing any novel objects. There was literally nothing. These animals were given the drug, and we just wanted to see if there was a signal, purely biological, having to do with sort of nothing else. So you're right. So this might provide evidence or might provide support for the fact that we really need to do context-dependent investigations, even with electrophysiology. Great question.
Okay, then, thank you so much. And this was the last talk for today. I want to remind you to vote for the awards. The ceremony is tomorrow. And yeah. Have a nice evening.
And hopefully see you at the party later.